Hello everyone. Today's our topic is how to make primers in silico in NCBI. So this is the homepage of the NCBI. You should go on all databases, go gene. Today we are searching a gene which is HBB. We should scroll downward. We are searching for a gene which is HBB, hemoglobin subunit beta in Homo sapiens. So this is the gene which is present in Homo sapien. We should open this one, HBB. Okay, this will take a time, so I have opened it earlier, which is here. As we see in the previous section, this is the summary of the gene. This is the genomic context, and today's our view is from this side, which is genomic region, transcripts and products. So this is the sequence of the gene. It has 1, 2, 3. We have primer. I am saying primer. We have 3 exons in this gene. So I am selecting this exon. Sorry. Zoom on range. It will take a little bit time. Okay, this is zoomed. Okay, now we should move it from here to like this way, and we will select, like we should take or select sequence after 15 to 30 base pair. Like we should select sequence for reverse primer from here to this, and then we should move here for our forward primer. So we are constructing our forward primer from here to this range. We should go on Primer-BLAST. So in this, they had selected already the range of the forward and the reverse primer, and also the, if you see here, the accession number, the sequence of this one. We are not going to change anything like but we should change this one. We should change, you should change the size of product size according to your own size, but I am selecting 200. TM temperature is selected min 57, 60, optimal 63. This is the uh, optimal temperature that we have. We are not changing anything, and we are going for get primers. Okay, it will take a few time, so I have performed it for you guys. These are the graphical views of the primer we have. Like this is primer 1, primer 2, primer 3, primer 4, primer 5, like 10 primers. If you wanted to see after graphical view, if you wanted to see the sequence and its information, it is given there. It makes 10 pair. The first pair of the primer is this one. This is the sequence of the primer. Here is mentioned the length, the starting and stopping point, also its GC content and temperature. I hope so you guys clearly know how to construct primer through NCBI. Thank you so much for watching.